Hello, everyone.
Hello.
Hello.
Hi.
It's nice to see everybody back in the cyber world here, so to speak. Now, one day it would be nice to meet you in person, right? It would be just nice to do that. So hello and welcome back. I hope you are well and continuing to be okay with the challenges from COVID.
Dr. Allen, this is Elena.
Hi, Elena.
Hi, I just realized I'm using a new device to log in. My computer died on me. So niece, that is me.
Okay, thank you for saying so, Elena, because I was starting to wonder about that.
Yeah, that's my daughter's short for her name. I have to figure out how to change the name at some point.
Okay.
I'll figure it out.
All right. No harm done. Thank you.
Okay.
Okay. So folks, we're going to wrap up chapter 20 today. Depending on how that goes, I may or may not get chapter 5 started. I don't need to start chapter 5. We are in good shape with our syllabus right now, with our schedule. Okay, so let me just point something out on the syllabus. I'm going to bring up my screen here for you. Okay, so here's the course syllabus for our class. Obviously, today is October 28th, right? So this is where we are. And by the way, hopefully you feel good that you really are closer to the end of your semester than the beginning. I know that's always something that students look forward to. Because, you know, let's be honest, you're working hard, you get tired, eventually we just need a break. So here we are, October 28th. Today I'm going to be continuing and finishing a lecture from chapter 20. On Tuesday of next week, or maybe today, depending on how things go, I will start chapter 5, okay? And I want to highlight that on Tuesday, I'm going to formally be assigning a homework problem to you from your workbook. That would be the same workbook that you had the unit conversions in, okay? So I'm not assigning it officially today. I'm going to be officially assigning it on Tuesday with the goal that you would do the homework next Tuesday and Wednesday and have it ready to discuss on next Thursday's class. But I'm going to highlight the fact that this homework is coming up. So if some of you want to get a head start on it, or you just prefer having a little bit more time to do these sorts of things, okay, please, you can start looking at workbook assignment number 3. And so let me bring that up, what exactly that is. Hold on a second. Okay, so here we have the workbook that you've all received a digital copy of. Okay, so I'm going to be assigning officially next Tuesday that you do assignment 3 for homework, which is this one right here. It's in page 8 of the handout, assignment number 3, antibacterial drug called APP. All right, so I'll page down and let you look at that really quickly. Here. There it is, okay? And so, you know, this is a data and graph interpretation homework assignment. And what you're going to find as we move deeper into the content for lecture exam 3, that part of the activities we are going to do are going to involve some more graphical interpretation of scientific data and some more math. Okay, don't be afraid. We're going to give you practice so that you can be successful. Okay, and so this homework assignment presents you with a graph of scientific data that's been plotted on this chart here. And I'm asking you a bunch of questions such as which axis is used to plot the independent variable. Okay, so you might have to do a bit of Googling for this. If you don't have experience with this kind of thing from other courses or other experiences you've had, you might have to do a little bit of Googling to try to figure out the answer to this kind of thing. So, you know, now you know a little bit even further out So if you want to start wrestling with this over the weekend, you could do that, okay? All right. Do you have any questions before we continue lecture?
For this assignment, did you want us to hand it in the Dropbox, or are we just going to go over it in class?
Okay, thanks, Samantha, for asking. You do not have to submit it. I'm not going to ask you to submit this work. Okay. We're going to go over this together in class next week. Next Thursday, we'll go over what you've done and make sure people are comfortable with being able to do this kind of thing, and you'll get lots of practice. But no, I'm not going to collect this for any kind of grade or anything like this. This is just to help you get more comfortable. Okay, other questions? Okay, so please understand what I'm saying here. Even though I'm not collecting it to grade, it is really in your best interest to try to figure these things out, because these kinds of things are going to be on your next lecture exam. Okay, lecture exam 3 will have data interpretation questions. Okay, let's continue here. All right, so last time we were getting started with Acyclovir. Okay, so Acyclovir once again is highlighted in figure 20.16. If you have your book open, or if you can get it open, that would be great. Figure 20.16. And we'll continue our Acyclovir story. Right, last time we spent basically the whole class listing out some examples of antimicrobial drugs. Most of them were antibacterial. I did give you an antifungal drug, which was ergosterol. Excuse me, it was amphotericin B. And I also gave you an antiprotozoal drug, which was quinolone. Okay, so now we're talking about an antiviral medication, in this case, a drug with the name of Acyclovir. And I'm just getting started here with this story. So remember, Acyclovir is a drug used to treat herpes viruses. There's two kinds that are typically treated with this kind of drug: herpes simplex virus, which causes genital herpes and cold sores, and herpes zoster virus, which is the culprit for chickenpox and shingles. Okay. So again, to understand how Acyclovir actually does what it does, you need to remember some important biology. So I'm going to ask you in the chat window here. Here we go. What is the basic building block of DNA called? And DNA is made of monomers of this thing that link together. What are these monomers called? Okay, thank you so much, folks. You're hitting it. Good job. They're made of nucleotides, right? So DNA is made of nucleotides, which is to say both forms of nucleic acid, DNA and RNA, are made of the basic building block that you know is a nucleotide, okay? Okay, remember that DNA and RNA are both made of nucleotides. Okay, let's continue. Another question in the chat window. Hold on a sec. Okay. There we go. What three components make up a nucleotide? What three components make up a nucleotide? Do you remember? Okay. Great job, folks. Good job. Okay. Even if you had to look it up or Google it a little bit, fantastic. You're accessing the right information, and that's fantastic, right? A phosphate group, a nitrogen base, and a sugar, a pentose sugar, right? A 5-carbon sugar. Okay. So that's also important. Remember that each nucleotide is made of 3 things: a sugar, phosphate group, and a nitrogen base. Okay. All right. So let's see, what else? What else do we need here? In terms of the biochemistry of nucleotides, the biochemistry of nucleotides is important so that you appreciate how this drug actually works, okay? Now, we're not going to go into heavy chemistry, but you need to appreciate a little something here, okay? A precursor to a nucleotide, okay, so a molecule that comes before the nucleotide. A precursor to a nucleotide is called a nucleoside. Okay, a precursor to a nucleotide is called a nucleoside. All right, so I'm going to ask you a question in the chat window for those that have access to their textbook. Okay, I'm going to ask you in figure 20.16 in panel B of the image. What color is the nucleoside? What color is the nucleoside? In panel B of figure 2016. Okay. Okay. So Chelsea, take a look there. Are you sure? Are you sure of your answer? Okay. It is purple, right? It's colored purple in panel B of figure 20.16. Okay. So that's what I want you to focus on for the moment, right? There are these precursor molecules to nucleotides, which are called nucleosides. So when a cell needs to manufacture a nucleotide, it starts with a nucleoside and it adds something to it, okay? And so we'll just break this down really quickly here, okay? A nucleoside has a sugar and a nitrogen base, just like a nucleotide, but lacks a phosphate group. It lacks a phosphate group, okay? Excuse me, I didn't type that into your notes correctly, okay? A nucleoside has a sugar and a nitrogen base, just like a nucleotide would, but it lacks a phosphate group. So a nucleoside is an unphosphorylated nucleotide, okay? Okay. So question in the chat window. What component of a nucleotide does a nucleoside lack? Okay, good. Good. It's the phosphate group. Awesome. Okay. Okay, folks. Now I have you focused on what you need to understand here. Okay, any questions so far? Okay, good. Let's continue. All right, so here's the deal. When cells need to manufacture nucleotides, what they will do is take a nucleoside and grab some source of phosphate, some source that has a phosphate atom, and they will link them together with a covalent bond. So basically, if you want to build a nucleotide, you grab onto a nucleoside and you attach phosphate to it, okay? To accomplish this feat of adding a phosphate group to a nucleotide, cells have special enzymes that do this. The enzyme that we're interested in at this moment for this conversation is called thymidine kinase. The name of the enzyme that cells use to add phosphate groups to a nucleoside, a guanine nucleoside. Okay, if you take a look at figure 20.16, look at panel A, upper left corner. Notice you see a molecule there, which is called deoxyguanosine. Deoxyguanosine is a guanine, because you see it there, right? Shaded in purple is a bunch of atoms with the label guanine. It is a guanine nucleoside. The name of the enzyme that the cells use to add phosphate groups to guanine nucleosides to make guanine nucleotides is thymidine kinase. Okay, thymidine kinase. All right. So some additional background here. Okay. Your human cells, our human cells, animal cells in general, have the thymidine kinase enzyme. And herpes virus in its own genome contains its own thymidine kinase gene. Okay, so human cells have thymidine kinase, and herpes virus also contains its own version of thymidine kinase. Okay, so folks, very soon I'm going to do a little bit of writing and drawing with the graphics pad, simply because it's going to be easier for me to communicate what I need that way. But let me say here, you know, I'm going to have to write a bunch of stuff out, so I'm going to use abbreviations and things to make sense of things. When I'm talking about a human cell thymidine kinase, expect that I will abbreviate it as TK. TK is going to be the human cell thymidine kinase. And when I need to refer to the herpes virus version of thymidine kinase, I'm going to indicate it this way with a little lowercase HV for herpes virus and an uppercase TK for thymidine kinase, right? So, you know, pay particular attention to that so you don't get confused about which version of thymidine kinase I'm referring to here. So human cells have their own thymidine kinase enzyme because human cells have their own thymidine kinase gene. Herpes viruses in their own DNA have their own slightly different gene for a slightly different version of thymidine kinase. Okay, and one thing that I should mention to you here is, you know, we know that thymidine kinase, because of what we said, its job is to link phosphate groups to nucleosides to build nucleotides. But part of our story here is an appreciation that the viral enzymes, when they do their work, they tend to do it faster. So what biochemists have measured in the lab is that the herpes virus thymidine kinase is 3,000 times faster than the human equivalent thymidine kinase. 3,000 times faster. What I'm basically telling you is, for every one phosphate atom that the human thymidine kinase adds to a nucleoside, the viral version has already attached 3,000 phosphates to 3,000 nucleosides, okay? That should be one of those, I'm watching a television show and I'm hearing ominous music kind of events here, okay? HVTK is 3,000 times faster than TK, okay? HBTK is 3,000 times faster than TK. Now, part of making new virus particles, as you might imagine, as you all know, because we've been through it in chapter 13, part of the process of making a new virus is to replicate the viral genome, whether it be DNA or RNA. So normally viruses will use the cell's machinery to accomplish that goal. In other words, typically if I have just an average Joe virus, the virus will go into the cell and it will use the human cell DNA polymerase to make copies of its DNA genome. Herpes virus is different. Herpes virus has its own version of DNA polymerase, just like it has its own version of thymidine kinase. So human cells have DNA polymerase, and as you know a little bit from chapter 8, there are actually many different versions of DNA polymerase in both bacterial cells and higher eukaryotic cells, but we'll just leave it simple right now and say DNA polymerase. So when I refer to human cell DNA polymerase in the future, I'm just going to do DNAP, okay? And herpes virus also contains its own version of DNA polymerase. So when I need to refer to herpes virus DNA polymerase, I'm going to refer to it as HV for herpes virus DNAP. Okay, so again, please key off on how I'm abbreviating these things so that you don't get confused about what molecule I'm talking about at any given moment of our conversation. Okay. Now, biochemists have measured the activity of the viral DNA polymerase and compared it to the equivalent human cell polymerases. And generally speaking, herpes virus DNA polymerase is about 100 times faster than the equivalent human cell enzyme. So HBDNAP is roughly 100 times faster than DNAP. Okay. All right. So let me just ask in the chat window, are you with me? Is everybody okay? Okay, anything you'd like to ask? Okay, all right. Okay, so let's see here. Okay, trying to figure out how to use my screen space most efficiently here because there's a lot to communicate. Okay, so let's begin here. Okay, maybe one more piece of important information for you. Okay. And I'm going to present it this way. What is the basic process that DNA polymerase enzymes use to link nucleotides together? Okay, that's a question that we need to be sure you're comfortable with. Okay, so what I'm going to do is describe to you how DNA polymerase links nucleotides together in a very basic, very skeletal chemistry sort of way, because you need to appreciate this to understand the beauty and nuance of how this medication acyclovir is able to work. Okay, so here's the basic idea, and please give me a little bit of artistic license here, so since I'm not much of an artist, but here's the idea. Okay, if we have a single strand of DNA, okay, so let's say we have a single strand of DNA here. Okay, and that single strand DNA is made of, a nucleotide linked to another nucleotide, linked to another nucleotide. Okay, is it just a chain of polymerized nucleotides? What does that kind of look like at the chemical level? Well, here's the deal. Okay, and again, I'm not much of a chemist, but I'm going to do this. Okay, I'm going to draw some of the basic chemical structure of a nucleotide using deoxyguanosine on figure 2016 as a guide here. Okay, I used to be able to draw this stuff by memory, but I'm getting old, so I can't do that anymore. Okay, but here's the basic idea. When you have a nucleotide, so let's say we have this. Okay, so here's the pentose sugar of a nucleotide, right? Pentose sugar of a nucleotide. Here we're going to have the guanine nitrogen base. Okay, I don't know if you understand carbon numbering schemes in organic chemistry, but the carbon that's immediately underneath the G of guanine is carbon 1. So I've got carbon 1, 2, 3, 4. Coming off carbon 4 of the ring, I'm going to have another atom, and that's going to be linked to a phosphate group. I'm just going to draw it like this, a phosphate group, okay? And then this would be linked to other nucleotides above, right? Nucleotide, so on and so forth, blah, blah, blah. Okay, and you'll notice that on the third carbon of the ring, traditionally you're going to have an OH group. Okay, so what I need to highlight for you is this. This OH group is critical for linking the next nucleotide. Critical for linking the next nucleotide, okay? So let me ask you in the chat window, does deoxyguanosine in figure 20.16 panel A have the OH group I have drawn in your notes? Do you see it? Compare what I've drawn, which admittedly is, my ugliness. Do you see it? Do you see the OH I'm referring to in your textbook? That's what I'm asking. Okay. All right. Okay, great. So if you appreciate that, then what you need to understand is that, if in your notes, this is the DNA chain that I've already got, and I want to link another nucleotide to it, then the incoming nucleotide, okay, I'll make this a different color. I'll make it a blue. The incoming nucleotide has to have this structure here. Okay, so this is going to be the incoming, meaning the next nucleotide is going to be linked to a pre-existing chain of DNA. Okay, the incoming nucleotide. So I want to link, in other words, in your notes, I want to link the blue nucleotide to the black nucleotide. That's what I'm trying to show you. And so the incoming nucleotide is going to link basically right here. This is where the attraction is going to be. And, I'm not quite doing it justice from a completely accurate chemical perspective, but I think this is close enough for you to see what I need you to see. Okay, so, all right. One more question, then we'll continue. Okay, hold on a second. Now, the last question I asked you was this. I asked you, does deoxyguanosine have the OH I have drawn in your notes? Now I want you to answer the same question for acyclovir. Does acyclovir have the same OH at the same location I have drawn in your notes? Do you see an OH in the structure of acyclovir in exactly the same location as deoxyguanosine? Okay, you don't see it there, do you? If you're really looking at this and you're getting what I'm trying to communicate to you, my point here is that acyclovir does not contain the OH group of a normal nucleotide. It does not contain the OH group of a normal nucleotide, and that's why this drug can actually be effective. Okay, so I'll just put this here in your notes and leave it as sort of a cartoon bubble. Okay. Acyclovir does not contain the OH of deoxyguanosine. That's the point that I'm trying to make. Sorry, that's kind of ugly. Folks, even though that is ugly, let me ask you. Can you manage to read these notes okay? I mean, do you think when you get them home and you're looking at them a week from now, you'll be able to decipher what I'm trying to say? Okay. All right. Thanks, everybody. Okay, so this is key, right? Normal nucleotides, like guanine nucleotides, have this critical OH that allows a normal nucleotide to link onto it. That's how you grow a chain of DNA. Acyclovir, as you look at your own textbook in panel A, as you look at the chemical structure of acyclovir, you notice that there is portions of acyclovir, particularly the nitrogen base, that really resembles deoxyguanosine, but below the blue shaded area in your textbook, you notice that acyclovir looks quite a bit different. It does not have a traditional sugar structure, and it certainly does not have that traditional OH that I'm highlighting in your notes. That is absolutely key to the behavior of acyclovir and what it is capable of doing to shut down virus replication. Okay, let's continue here. All right, so I'm going to put that to bed and go to the next page. Okay, so what we'll do now, that was bullet point number 8. What we'll do now is make a basic schematic of how cells, viruses, and acyclovir interact. Okay, and so to do this, I'm going to go back to drawing. It's just easier for me to communicate it this way. All right, so I'm going to split the screen into three columns here and kind of do it this way. So we'll have a column here. We'll have a column here and the third column. Okay, that should suffice, I think. All right. So in the first column, I'm going to show you what happens inside a normal cell that's uninfected. There is no virus and there's no drug. Okay, so we're going to have an uninfected cell, no virus, no drug. Okay, so the basic idea of this, when a human cell needs to replicate DNA, what that human cell is going to do is going to take the deoxyguanosine nucleoside, it's going to link a phosphate group to it through the activity of thymidine kinase, right? TK, that's going to be the enzyme, and you're going to get a mature guanine nucleotide. Okay, so you take deoxyguanosine, which is the nucleoside, you link phosphate to it through the activity of the thymidine kinase enzyme, and you build a mature guanine nucleotide. That mature guanine nucleotide can then be used to make DNA. In the basic process that we just schematically diagrammed out here for you in the page above these notes. Okay, so that's your typical everyday, this is what your cells do. Okay, now what happens when you have a herpes virus infection? Well, okay, so we're going to have an infected cell. That means herpes virus is present. But right now in our conversation, we're not going to have any drug. There's not going to be any Acyclovir in this middle column. Okay. So, all right. Boy, I wish I was on a whiteboard. It would be so much easier to do this. Okay, I'm going to change colors simply so you have a better way to distinguish one column from another. Okay, it's just so hopefully it's easier for you to read these notes. Let me go with red here. Okay. The only reason I'm choosing a different color again is to distinguish this stuff in your notes so you don't get confused. Okay, so what you're going to have is the cell. You're going to have deoxyguanosine plus phosphate with thymidine kinase go to a guanine nucleotide. At the same time, you're going to have the same chemical reaction taking place with the activity of the viral enzyme. So this will be deoxyguanosine plus phosphate plus HVTK, and that will build a guanine nucleotide. Okay. I should have put in the previous column, and I'll go back and fix that now, that when the cell makes DNA, it's going to use DNA polymerase, right? DNAP. Let me ask you a question in the chat window. Here we go. Is there a chemical difference between guanine nucleotides made from TK versus herpes virus TK? Okay, thanks folks for participating. You know, you have to 50-50 chance of getting this right, correct? So the question is this: is there a chemical difference between the guanine nucleotides made from human cell thymidine kinases versus herpes virus thymidine kinase? And the answer is no, there is no difference. There is no difference. The cell does not know the difference between a guanine nucleotide made from one of its own enzymes and a guanine nucleotide made from a herpes virus enzyme. The cell cannot tell the difference. There is no chemical difference. They're the same nucleotides, okay? It's just that the herpes virus enzyme is faster at doing its work. Okay, let's continue. All right, so you're going to have the next stage that will happen, and sorry, this is so cramped for space. I dislike this, but okay, you're going to have the cell building some of its new DNA with its DNA polymerase. So the cell makes its own DNA with its own DNAP. All right. And at the same time, the herpes virus DNA polymerase is going to make herpes virus DNA with the nucleotides that are available in the cell. The virus inside the cell makes new viral DNA using HVDNAP. Okay. Now, question in the chat window. Which DNA polymerase is faster, the cell version or the virus version? Okay, it's the virus version, right? It's the virus version. Think about it, folks. If you have, all right, let's think about this logically for just a moment. The human cell has billions of nucleotides of DNA spread across 46 chromosomes that have to be copied so that I can make a new cell, right? So there's a lot of DNA to be copied in one genome of one human cell. The herpes virus genome is paltry. It's on the order of tens of thousands of nucleotides. So there's very little DNA to build new virus particles. So that means that in terms of competition here from the human cell DNA polymerase versus the viral DNA polymerase, the viral DNA polymerase is faster and it has less nucleotides to polymerase to make a new virus than the human enzymes have to achieve. So long story short, most of the DNA synthesis in a virally infected cell is going to be DNA synthesis that supports replication of the virus instead of replication of the cell. Okay, so question in the chat window. Am I making sense? Okay, so take-home message here is that most of the DNA being synthesized in a herpes virus infected cell is actually going to be DNA to support new viral production rather than new cell production, okay? Most DNA being made will be herpes virus DNA. Okay, that's the problem. The infection of your cell with a herpes virus, because its enzymes are faster and there's a smaller genome that needs to be copied in the first place, means that the majority of DNA being synthesized in the cell is going to be viral DNA instead of cell DNA. So basically, the virus has turned the resources of the cell to its own advantage and set up a manufacturing toy shop at the North Pole to make viral toys, right? Santa Claus can't make the natural toys he wants for the children. He's got to make warped infected toys, okay? All right, so that's what's going to happen inside the infected cell. All right, now, here's where we get to the very nice story with the Acyclovir and the Achilles heel situation. Any questions so far? Okay, so now we have an infected cell. And we're going to have herpes virus present. Plus Acyclovir. Now, a lot of what I could say would be a repetition of the middle column, so I'm not going to try to write that down and just give you an ugly presentation. But what I want to focus on is, well, how does Acyclovir interrupt this story of the middle column? How does Acyclovir disrupt what the virus is doing? And so let me ask you a couple questions in the chat window. Whenever you as a human being have attempted to do anything too fast, what usually happens? Whatever it is that we're talking about, writing a check out to pay a bill, writing an answer down on an exam, playing with your children in some way, right? The faster you try to do something, typically the more prone you are, and me too, of just making a mistake, screwing it up, okay? So let me put this idea down in your notes. Speed leads to mistakes. Speed leads to mistakes. Now let's think about that. We've gone to some pain here to define for you the fact that the viral enzymes are faster than the human cell enzymes, right? The viral enzymes are faster than the human cell enzymes. That is the Achilles heel of herpes virus. Its enzymes work too fast to be selective. Its enzymes work too fast to be selective. Okay, so let me just put these ideas down. HV enzymes work too fast to always be accurate. Okay. All right. So let me use a ridiculous analogy with you at the moment. Okay. Let's say for a moment that we are all going to go to an Easter egg hunt. We're going to go to an Easter egg hunt, and so we're at a local park, and before the Easter egg hunt has been, before the Easter egg hunt starts, okay, Tiffany, see you later, good luck. Before the Easter egg hunt starts, you know, a bunch of adults placed Easter eggs out into the lawn for a bunch of toddlers to go out and have fun gathering Easter eggs, okay? But stay with me here on this, okay? Here we go. Will it be fair if a bunch of teenagers show up to an Easter egg hunt for 3-year-olds and they want to gather eggs too? Is that going to be a fair thing? I mean, I don't know too many parents that are going to be happy with that scenario, right? If you're a parent and you brought your child to have fun at an Easter egg contest and a bunch of obnoxious teenagers show up and they grab all the eggs, you're going to get pissed off. Okay, that's a jerk thing to do. All right. But that's basically what's happening here in terms of the herpes virus. A bunch of jerk teenagers showing off, showing up and grabbing all the phosphate atoms and things like that that we need to build nucleotides. Okay, so keep that idea in your mind. Now, what I'm about to say is in the context of this silly analogy. I'm not saying anybody should ever do what I'm about to say, so don't, take it as a lighthearted thing with my analogy here. Okay. And I'm going to type this into the chat window. Do you think we could even the playing field of teenagers versus 3-year-olds if we insisted that the teenagers drank a bottle of wine before participating? If we got the teenagers drunk, would the 3-year-olds have more of a chance to at least get some eggs? Okay. I hope you can see in my silly analogy, because I'm not suggesting you get teenagers drunk, is that if we did something to disrupt the teenagers, then maybe the children would have a little bit of an extra chance here. And so where I'm heading with this is that, if we had some drunk teenagers going off competing against the children, I bet you when we come back and everybody opens their bag and looks at their eggs, next question. Since the teenagers are drunk when hunting for Easter eggs, do you think they might accidentally grab some rocks and place them in their bag? You got to use a little bit of artistic license here with this silly analogy, but this is the point I'm trying to make. The teenagers are drunk. They're going to go out there and race around just to be obnoxious to the children. Well, they're probably going to grab some round rocks that kind of have the overall shape of an egg simply because they want to win. So they're being overly aggressive, overly fast, and because they're drunk, they're not going to be very selective in what they grab. Okay, so this is the basic idea I'm trying to plant in your mind in terms of how Acyclovir works. With Acyclovir, okay, here we go. Acyclovir is a false nucleotide. It is a false nucleotide. Okay, you can see that because I pointed out up above here with the green lettering that acyclovir does not contain the OH of deoxyguanosine. It does not contain the atoms needed to link another nucleotide into it. So what that means is the following. Okay. The virus can attach acyclovir to the end of a piece of viral DNA, but you cannot add an additional nucleotide after it. Oh, sorry, this is getting ugly here. Okay. Okay, let me read this again. The virus can attach acyclovir to the end of a piece of viral DNA, but then you cannot add an additional nucleotide after it because the acyclovir lacks that special OH that I pointed out up here. Okay, so I'll just put that down in parentheses again. Acyclovir lacks an important OH group, right? And think about the notes above. Here's the deal. Human DNA polymerase may be slower than the viral enzyme, but because it's slower, it's also more selective. So human DNA polymerase can tell the difference between a normal nucleotide like deoxyguanosine, and it will never grab onto acyclovir because the human enzyme knows that acyclovir is not the right thing to link into its DNA. So the human enzymes will do their work slower and more correctly. The viral enzymes, they are faster. They're drunk. They're sloppy in what they're doing. And so the viral enzyme will look at acyclovir, and because it resembles a nucleotide, it will grab onto acyclovir, link it into its own DNA. But when it does that, bam, we've got the virus. Because as soon as you link acyclovir into the DNA on the bottom end of acyclovir, you do not have the correct atoms, the OH atoms that I'm pointing out. You do not have the correct atoms to add the next nucleotide. So in other words, the presence of acyclovir blocks subsequent DNA synthesis of the viral genome. Okay, acyclovir blocks additional viral DNA synthesis. Okay. So let me ask a question in the chat window here. Does acyclovir represent a cure for herpes virus infection or simply a way to control the infection? Okay, good job, folks. You can see the difference, right? Acyclovir is a magnificent way of suppressing viral replication because you're blocking viral DNA synthesis. However, it does not remove any of the original virus particles that happen to be in your body. So acyclovir and many of our antiviral medications are very good at suppressing the virus replication and therefore preventing significant disease consequences from that. But they are not cures because they can't rid the cells of the body of the original virus that are inhabiting your body. Okay. All right. So that's a bit of a long-winded story, folks. I hope I made sense to you. Do you have any questions? Okay. All right, gang. So there you have a conversation about acyclovir, and we'll use acyclovir as our case study of an antiviral medication. Okay. If you don't have any questions about that, I will continue. All right. So, so we've done, I hope, a fairly decent job of summarizing some of the major ideas of chapter 20. We could talk about one more topic here with the remaining time that we have. Okay, and that would be, so we have all these drugs, there's just almanacs and databases filled with, medicinal chemistry, molecules and things like this, and we're always making new ones and things. So let's talk about the human struggle for alternative antimicrobial drugs, right? What are we going to do? We know we've got a problem with, resistance, antibiotic resistant strains of bacteria. Viruses mutate and become resistant to certain medications. This is a battle that's going to be till the end of time, right? We're never going to actually have a final complete victory over this situation, not as long as Mother Nature has a say in it. So let's talk about the human struggle for alternative antimicrobials. Okay, so let's talk about ways that humanity can continue to try to stay ahead of evolution in terms of, trying to combat the fact that as soon as you put a drug on the market, microbes are evolving a solution around that drug. Okay, we said earlier it takes, on average 10 years, if not a little bit longer in some cases, to get a new drug to market. You get it to market and then you expose it to a community of people with a community of microbes, and the microbes start mutating their DNA, changing in ways so that they resist the medication. So one way humans have done, and I think comes out in the language of this chapter, is to take existing drugs that have been somewhat successful and tweak them or chemically modify them. So one way to deal with this issue of drug resistance, and maybe I should put that down up here, is the following ideas. Okay, first off, take existing drugs and modify them. Okay, that almost always involves a team of organic chemists and or biochemists working at a pharmaceutical company. This is the domain of organic chemists. And you can bet that a place like Pfizer or Merck or GlaxoSmithKline, they hired tons and tons of organic chemists. As you read through this chapter, you read about the penicillins, and that there's many different varieties of penicillins, right? Some of those varieties are semi-synthetic, meaning the drug companies started with the original penicillin found in the mold of Alexander Fleming, and they started to chemically tweak it in the lab. And some of those drugs actually make it to market and have different therapeutic profiles. Okay. Well, a second way of dealing with drug resistance is to identify new drugs from new sources. Okay. Okay. There have been some projects out there in the United States where undergraduates actually have been asked to participate in this project, where undergraduates go around their local communities and collect soil samples from the communities they live in. So the idea would be, let's get a soil sample from Lancaster County. Let's get a soil sample from York County, from Dauphin County, right? You get as many soil samples as you can from around the world, and you use these soil samples to discover new bacteria, new microbes of any kind that might in fact be a source of a new antibiotic. We simply need to find these things. They're out there in Mother Nature, so how do we find them? Okay. Ask humans to help gather random soil samples from around the world. These samples may contain new sources of antibiotics. Okay. Well, there are new categories of drugs called antisense drugs. Okay, antisense drugs. Antisense drugs are made of nucleic acids. And so let me ask you this. Let me ask you a question in the chat window. When a ribosome translates messenger RNA, is the messenger RNA single or double-stranded? Okay, gang, very good. It's single-stranded. It's single-stranded. In other words, the messenger RNA must be single-stranded if the ribosome is going to decode it into a protein. Well, what if we put pieces of DNA and or RNA into the cell that could actually bind to the single strand messenger RNA and turn it into a double strand molecule? What if we could do that? Well, I think you can appreciate if we had the ability to do that, and we have some ability to do that, then we could block ribosomes from making proteins. Okay, Samantha, hold on a sec. Okay. Yes, I do mean May. Thank you. Sorry about that. So antisense, right? Add complementary fragments of DNA or RNA to a cell that turns single-stranded messenger RNA into a double-stranded molecule. This blocks ribosome activity. If you could achieve that goal, and it is technically daunting to do, but if you could achieve that goal, you would definitely have the way to disrupt a cell. You could disrupt a fungus, protozoan, bacterium, and kill it, because the ribosomes couldn't make the proteins and enzymes that the cell needs. Okay, so that is a strategy that scientists have been working on. Another therapy, another strategy for dealing with this resistance that companies are working on is something I alluded to before, either in lecture or lab, and that would be the topic of using phage therapy. In other words, using bacterial viruses to kill pathogenic bacteria rather than using traditional antibiotics like penicillins and so on and so forth. Okay. So perhaps bacterial viruses would be better at killing bacteria than some classical antibiotics. Okay. As I said, that has been used successfully in Eastern Europe. I think there's a whole host of reasons in the United States, from economic and political reasons, of why we haven't really seen that in the US. And, you know, phage therapy is interesting, but it's not it's not a panacea. I mean, as soon as you start killing bacteria with a bacterial virus, other members of the bacterial species is going to start to evolve to try to survive the phage. So there's never a perfect solution, right? And so what I'm talking about here is that evolution, which is Mother Nature's way of introducing purposeful genetic change to allow a cell or a virus to survive some kind of threat, is always at work. So there has been some attempt in research labs to find ways to actually slow down the mutations process itself. What if we could slow down or retard evolution? We might be able to slow down how rapidly microbes become resistant to drugs. Can we slow down evolution itself? Labs are working on this. Okay. Another thing we could do. One thing we haven't talked about here, but which is hinted at on figure 20.23 of your chapter here, chapter 20. Take a look at figure 20.23. Notice the drug synergism picture. And the idea here is that maybe, just maybe, there's one medication that will not kill a bacterium, and there's a second medication that will not kill a bacterium. But for some reason, when those two medications are present in the same moment, in the same cell at the same time, all of a sudden the cell dies. So there's some kind of chemical interaction or kind of cumulative teamwork effect of having 2 or maybe 3 or maybe 4 different combinations of drugs together at the same moment. If we can identify those sorts of interactions and catalog them in the science lab, we might be able to combat some levels of resistance that way. Okay, so what if a combination of at least 2 different drugs is able to kill a microbe together instead of either by itself? Big companies are working on this. And when I say working on this, you will find big companies that, they have huge facilities with just rows and rows of robots working tirelessly to take different medications and drugs that we've purified and mix them together and then assay them, test them to see if the combination at different concentrations of drugs are capable of doing anything meaningful to pathogens we're interested in destroying. It's a tough process. It's slow and tedious, which is why it is perfect work for a robot. Okay. All right. All right, folks, I think I'll leave it rest right there. Okay, we're about a minute early, but I don't think makes much sense to continue from this point. Do you have any thoughts or questions right now about anything we've talked about in chapter 20? Okay, then gang, let's take our standard break here. We'll start lab at 2 o'clock and just talk about a couple different things we need to talk about this afternoon. So take a little break. The Zoom channel will stay on. You don't need to log off. I'll see you in 15 minutes, okay? See you soon.